And as my supervisor has once put it, Dr. Brennan Gerd, there's no one who likes to talk science more than Chris Perry. So what better guest to have on to chat about science with me today? Dr. Perry, if you wouldn't mind giving yourself an introduction and some of your research and even some of your academic background and what got you to the point you're at today. We both went to the University of Guelph. You were there for a lot longer than I was, but I did do my undergrad there. So we have that.
Oh, which degree did you get at Guelph? HK? Human kinetics?
You would think, considering I'm in an exercise physiology PhD program, but no, I was in the botany program.
Oh, very cool. Very cool. I've been saying lately I need to learn a lot more about plant metabolism, but I don't know why.
There's a lot of overlap with plant metabolism and human metabolism. Like, I feel like I had a good biochemistry understanding in order to apply it to humans. So it didn't set me up poorly.
Absolutely. An entirely separate chat. I think we need that one. No, I'm happy to do this. Thanks very much for the suggestion, the invitation. I'm an associate professor at York University in Toronto in the School of Kinesiology and Health Science. Our lab examines the role of mitochondrial stress, if I could use that term broadly, as a potential contribution to muscle weakness in various diseases. The background was, as you noted, University of Guelph, so bachelor's, master's, PhD at Guelph. Very briefly, I think I got into research because of Dr. Brian Wilson. He was my mentor for a fourth-year research project, whole body applied physiology, no muscle biopsies, no mitochondria. It was all respiratory gas exchange with exercise and various interventions, which are actually pretty fun and pretty cool. And then I did a master's with him, again, applied whole body physiology. Actually, we were looking at the effects of breathing 60% oxygen every single workout to try to increase the amount of wattage, the workload that you could achieve. So the whole idea was if you could work at a higher intensity each workout by breathing supplemental oxygen, are you essentially training harder, independent of any direct effects of oxygen, and can you become more fit? And that was all gas exchange. And then I did a PhD with Lawrence Spreet, which absolutely opened my eyes into how skeletal muscle metabolism is regulated by key enzymatic steps and substrate transporters. Again, all in a human exercise context. That was with muscle biopsies. And then I became interested in, essentially, I think I have a pretty good handle on how healthy muscle adapts its metabolism to chronic exercise and becomes better, to put it simply. I became very interested in, what about conditions that go the opposite direction? So in other words, disease. So then I did a postdoctoral fellowship for a few years in insulin resistance, essentially. So how overeating and sedentarism can let's call it, create mitochondrial stress in relation to dysregulation of whole body metabolism. Then I became faculty. I went, let's do muscle weakness disorders. No, not as nonchalant. I just became very fascinated with, what about diseases where exercise as a therapy might not be so feasible? What about diseases where there might be so much muscle weakness it becomes difficult to engage in exercise at an intensity that might actually be beneficial? Now, that's not always true for all muscle weakness disorders, but now here we are. We're studying how diseases of a variety of different primary causes that have nothing to do with mitochondria actually eventually lead to mitochondrial failure, and how that can now be a secondary contribution. And so we're looking at Duchenne muscular dystrophy. We're looking at cancer cachexia and some other diseases as well. And through that, we're looking at nutritional, physical activity, and pharmacological therapies to try to rescue mitochondria as a combinatorial approach for treating muscle weakness. Most of our work right now is partnering, let me say it differently, most of the therapy-driven work, separate from the mechanistic discovery, is partnering with companies that have very novel mitochondrial enhancing drugs that will go in the mitochondria and preserve and shield them from stress to try to rescue muscle function. And in some cases, in some diseases, just by targeting the mitochondria, we can actually preserve some muscle health. So it's been my early roots in the regulation of skeletal muscle metabolism with exercise gave me a foundational understanding of how muscles respond to the stress created by exercise, and has really carried over to compare to the stress created by disease. And so I'm sure we'll get into that a bit more. But I think for students, whatever you study, even those in the exercise field, you can get clues by studying the disease literature and vice versa.
Yeah, every single topic in your journey there is so interesting on its own. I wanted to interrupt you and be like, so if we breathe 60% oxygen, what happens to our fitness?
Yeah. What
were your results from your master's?
For that, we saw greater improvements in I got to go way back to that. That was 2003. Oh my God, it's 2023. Don't say that sentence. There were greater improvements in time trial to exhaustion at about 90% VO2 max. But a follow-up study we didn't see that effect again. And so while I moved on to different directions, one idea that came from that was it might actually depend on the initial fitness level of the participant. And so I can only speculate about whether it will or will not work. But follow-up studies, other studies in the literature have pursued that question in various populations, including, I think it was people with diabetes. And yeah, sometimes it improves a fitness measure and sometimes it doesn't. But I think there's a lot of opportunity to talk way more than I should here about why that is. It's all speculative. I think in our case, it would be really interesting to do a study to see if participants of average to low fitness really aren't going to benefit from a slightly higher workload. Just the fact that their exercise training period is already a new stress to their muscle, that adding a few more watts with hyperoxia is a drop in the bucket. And so maybe hyperoxia, this is purely speculative, maybe hyperoxic training would be more likely to improve fitness in people who are already training and just need to get a slightly higher workload in their training regimen. And I haven't actually searched in about 5 years if anyone's done that study, but that's where we left off as speculation, and maybe the differences in our studies were due to some fitness differences. So
yeah, that's super interesting.
Yeah.
But I have diverged the conversation because we are here to talk about mitochondria. So initially, why we even connected on this is because I put out a tweet asking, is there any mechanistic data to suggest that zone 2 training would promote greater mitochondrial adaptations or improve mitochondrial function when compared to higher intensities of exercise? And because I'm just kind of confused as to these, like, what mechanism would drive greater adaptations if you're working at a lower work rate than if you were just doing it at high intensity exercise? Because we know your work has shown that you can improve aerobic capacity and improve these markers of aerobic metabolism through high intensity interval training. So that's where I was like, my mind is confused on this whole zone 2 argument. But to even take a step back, when you're in the lab studying mitochondria, like what is the laboratory protocol for measuring mitochondrial function, and how would we differentiate mitochondrial function from improvements in mitochondrial content? Like if we're measuring markers of mitochondrial biogenesis or density, what can those be different from markers or improvements in mitochondrial function?
Yeah, in terms of how it's measured, it's evolved over the years. And so you'll see a lot of papers now using technologies that weren't as commonly used. They were present, they were available, but they weren't as commonly used in the classic literature that really started to pick up in the 60s and 70s and really exploded in the right up to the 2000s. So now, there's more and more studies using respirometry, which is essentially a small glass chamber that you can put your tissue sample in, whether that's an isolated mitochondria or various preparations of actual tissue that still have their mitochondria. And the chamber has an oxygen sensor, really no different than an oxygen sensor in the car. That might not make sense. Maybe that's a bad analogy. And it's actually monitoring the amount of oxygen in the liquid, in the buffer that your sample was placed in the sealed chamber. So as your mitochondria make ATP, they will use oxygen to, as part of oxidative phosphorylation, to break substrates down, whether it's glucose or fat or amino acid derived substrates. So the very fact that mitochondria are using oxygen to make ATP means we can monitor how much oxygen they are using in the chamber by monitoring how quickly oxygen disappears. So when you see a respiration graph in a paper, that it's actually monitoring how quickly oxygen has left the chamber, or rather been consumed by the mitochondria. So you actually monitor a decline of oxygen consumption. And if you start adding to that chamber carbohydrate-based substrates or fatty acid-based substrates or amino acid-based substrates, you'll actually see the oxygen drop faster and faster and faster because the mitochondria is using that oxygen. That's respirometry. Respirometry has been around since the early 1900s. But in the 60s and 70s and thereafter, some studies were using that approach. It's far more common now, but the limitations of the approach were addressed by using other methodologies that, from what I can tell in the exercise field at least, is really starting to decline, which is unfortunate. Because when you open a basic textbook on how skeletal muscle metabolism is regulated, let me say it more precisely, how mitochondria are regulated. And what does that mean? How does a mitochondria choose to use carbohydrate, choose to use fat, choose to use amino acids? Well, there's either transporters or enzymes. And so when you unpack what these pathways are, you start to answer your question, how does one measure mitochondrial function? Well, what aspect of function do you want? If you want ATP synthesis, the question becomes which fuel is being used to measure that. Well, then how do you measure glucose oxidation? How do you measure fat oxidation? Classic approaches, and there's a wide variety there, would trying to avoid getting too deep into the technical aspects, but there'd be a combination of approaches that didn't use respirometry per se. One would be looking at the degree to which rate-limiting enzymes were activated. So, for example, glucose oxidation is regulated in part by how activated pyruvate dehydrogenase can become. And so that was often performed with an in vitro assay where you essentially run a PDH activity assay. However, very careful attention would be needed to be placed on whether or not the process of freezing and homogenizing that muscle trapped PDH in its activated state relative to its inactivated state, which is largely mediated by phosphorylation. So there are chemicals you can add essentially to your homogenate that prevent your pyruvate dehydrogenase from changing its degree of phosphorylation. And now when you run the enzyme assay, you can actually say, oh, I'm not just running an activity assay that tells me how much of the enzyme is present. It's not a total PDH activity assay, it's an activated assay. That was used as one of many tools to start to get more insight into whether or not exercise would promote carbohydrate glucose oxidation or not. However, on its own, that's just one enzyme. You have no idea if from that alone, if there was more or less glucose oxidation. So when we say, to answer your question, how does one measure mitochondrial function? We have to be very clear. I think we're talking about ATP synthesis. And then at that point, are we talking about which fuel? So you're still left, even when you run an assay targeted at a rate limiting enzyme in any of these pathways, with wondering if did that actually mean that the whole system, all the pathways, all the transporters, and even before the muscle, the delivery of that fuel in the blood created a net sum change to the oxidation of that substrate? So what other approaches can you do? Well, a lot of groups were using radioisotopic methodology, where they would take a tissue sample and they would provide a radiolabeled glucose or radiolabeled fatty acid to a mitochondrial preparation from that tissue, and open the textbook, a biochem textbook, and look at, okay, if I've labeled this carbon in glucose or this carbon in fat with a radioisotope, what do we know from classic biochemistry of where that carbon winds up as a product of the reaction? So very common, very often people might monitor carbon dioxide production in response to a substrate, but look at how much radioactive carbon dioxide was being produced as an example, and then that is a direct product of oxidation, the use of a fuel to make ATP. Now that is not targeting a single enzyme, that's targeting the whole machinery within the tissue. So when you combine that end output measure of a product of oxidative phosphorylation, or let me use that more broadly, substrate catabolism of one fuel, compare that outcome measure to targeted assessments of specific rate-limiting enzymes, like we used an example previously, you start to get a much more in-depth perspective than I think what is happening with the popularization of respirometry. And this is just my opinion, but the problem, and I use respirometry, we all do, but we need to be very aware of how we're treating the tissue sample. The question remains, whatever we're assessing, did we trap the state of all these pathways as it existed in vivo? And those were key questions that drove methodologies for decades with these other assays to measure, as we're putting it, mitochondrial function. But now that respirometry has emerged, and again, we don't want to get too much into the details here. I could use some if you wish down the road, but my advice to students reading this literature is to read the methods section. Ask yourself, when we measure respiration to measure mitochondrial function, what does that mean? What substrate was provided to the tissue sample, to the mitochondrial preparation? Was it a carbohydrate-based substrate? Was it a fatty acid-based substrate? Was it an amino acid-based substrate? Because to say respiration changed as an index of mitochondrial function is incomplete. Which substrate adapted to the exercise stimulus or the training stimulus, depending on the study design? Did this study look at a glucose-based substrate? Did it look at a fatty acid-based substrate? Did it look at an amino acid-based substrate? If so, then the conclusion is constrained by that design in the methodology section. But now, the last point I'll make here is now what that means is we have to think very carefully about what rate-limiting enzymes were, not just enzymes, potentially transporters, were being captured by the design of that substrate protocol. And then we have to think, this is the way we did for decades, how did the tissue preparation procedure retain the degree to which those rate-limiting enzymes were activated or inactivated? And that's what's missing in most of the respirometry literature. So how does one measure mitochondrial function? Those were the combinations historically, at least in tissue-based samples. And that's a summary of what's being done now. But I think careful considerations that were done for decades with these other techniques are not always being applied to the popularization of respirometry. So while that doesn't give you a definitive, a simple answer, the point is exactly that, to be very careful, especially as students delving into this literature or new investigators new to the area, of there's several ways to put this, but of oversimplifying discoveries when we need to actually pay way more attention to the method section of papers, in my opinion, combined with self-teachings of classic metabolic control theories that, quite frankly, I think are being not fully integrated into the discussion of a lot of papers. And I don't mean that as a negative criticism. I mean it as a guidance to people getting into this field to don't just look at the conclusion. I think there's a huge opportunity to integrate the popularization of respirometry with classic theories that are just a matter of sitting down and learning this stuff. So again, it's all coming back to your question of how does one measure mitochondrial function? Well, what do you mean by function? What function are you interested in? You know, not you personally, but
Right.
the individual. And it has to come down to what substrates are you targeting and what's rate limiting. And all of this is an answer within tissue lysate. Ultimately, combining it with an in vivo whole body assessment of substrate oxidation is critical. And very popular with that is gas exchange with RER and so on. And far more advanced approaches would be in vivo radioisotopic approaches as well.
Right. And like, is there any situations where these outcomes disconnect? Like, would you ever see a downregulation in PDH, but you see an improvement in these glucose-based respirometry outcomes? Like, do they usually correlate with each other, or do you have to measure the enzymes or the transporters with the respirometry results as well?
Classically, changes at the mouth with gas exchange would be related to a well-controlled, properly designed assay for, say, pyruvate dehydrogenase activity in a muscle biopsy. That's been shown that the degree to which PDH is activated in skeletal muscle in response to, say, an exercise stimulus is related to the gas exchange we see at the mouth.
In what way? Like, specifically?
Yes, so let's say as you begin to exercise at increasing intensities and you start to see your RER go up, which is indicative of a transition towards carbohydrate oxidation, studies have shown that is related to a muscle biopsy-based assessment of pyruvate dehydrogenase activity.
Okay.
And that's the kind of combination that becomes very insightful when the design of the in vitro assays are carefully considered. Did we truly trap? Did we design the homogenization approaches to truly trap the degree to which that enzyme of interest was activated? To answer your question in regards to respirometry, that becomes a bit challenging. Off the top of my head, I can't think of a study. I'd have to do some searching that compared RER to mitochondrial, say, pyruvate oxidation. I'm sure it's out there and I'm sure I've read it. It's just here I am blanking on an appropriate reference. But nevertheless, what I can say is that level of consideration of when we take the biopsy and then when we prepare our muscle sample to measure pyruvate oxidation, pyruvate-driven respiration in respirometry, what is in our buffers when we prepare the muscle to trap PDH in its activated state? And the answer is nothing. And that's where there's enormous opportunity for the respirometry field to hit pause. And this doesn't just apply to exercise, it applies to our research and everyone's research in any field, to hit pause and go, how can we advance this field of respirometry? When I say field, I don't mean exercise, I mean the global field. What can we do to hit pause and advance our methodologies? And quite frankly, the lessons are there from the past on non-respirometry based assessments. You know, what were these chemicals we were adding to trap PDH's phosphorylation state? I keep coming back to it as only one example. There's many different enzymes to consider. Well, we can debate, should we? I think rather the question is, why would we hit pause and start to consider this? Because I think the elephant in the room that's not being stated is when you take a biopsy, when you remove tissue out of the body, things start to deviate from the way they were. When you homogenize, now you're deviating things even more. You've destroyed the cell membrane. You've replaced the cytoplasm with whatever buffer you've had, which ideally is designed to reflect osmotic balances of the cytoplasm and other aspects, but it won't perfectly modify it. And most importantly, you've now removed the stimulus. If we're talking about a biopsy after exercise, you need to generally freeze that instantly. You want to plunge that biopsy needle into liquid nitrogen so that the enzyme stays trapped, that phosphatases and kinases do not change it or other forms of regulation. But we don't freeze when we take a biopsy prior to respirometry, because freezing actually forms ice crystals and punctures the mitochondrial membranes and destroys them. And with respiration, you want to keep your mitochondria intact because they, again, a lesson for another day, ATP synthesis, if you want to assess oxygen consumption as an index of ATP synthesis, what we're talking about is not an isolated enzyme activity. We're talking about an intact phosphorylation system, the entire electron transport chain, ATP synthase, and everything else upstream. All of those processes are driven by membrane potential. And as soon as you freeze a sample, you create daggers of ice, essentially, that are going to puncture anyone who's ever done freeze-thaw cycles of a sample to optimize protein yield in homogenization will appreciate this, that if you want to measure an intact the function of intact mitochondria, the last thing you can do is freeze. But now you have a problem. If you're not freezing, you're not trapping the enzyme of interest in its degree of activation. So what do? Well, currently, and as has been the case for decades, you take the muscle out, you put it in a dish or a tube with buffer that's designed to keep it essentially alive, proper osmotic balance, proper salts and other things in there, and you put that tube or dish on ice. Now, you don't freeze the liquid, but you keep it super cooled because the cooling slows down the reversing of whatever regulation had occurred in vivo in the body in response to the stress. But that stress, that exercise stress, if we're talking about an exercise study, is no longer occurring in the dish. So we run the risk of actually what we're interested in to measure to explain a change in glucose oxidation at the mouth or a change in fat oxidation in the mouth. That the study was designed to say, look, if I did this exercise intervention and I see with respirometry, with whole body gas exchange, there was a change in the degree to which I was using carbohydrate versus fat oxidation, is that due to this enzyme or this pathway in the muscle? Well, as soon as you stop exercising, you remove the stimulus that caused the altered regulation of those enzymes. So when you remove the biopsy, that activation by exercise, that stimuli from exercise is gone. So these pathways will reverse. And the way to slow that down but not stop it is to cool your tissue. Ideally freeze it, but if we want to measure mitochondrial function, we can't freeze. We're going to destroy the mitochondria. So we have to cool it, but the cooling is not perfect. And so, yes, I'm getting nitty gritty, but the point of this is, as I understand it, is to really provide some key considerations for anyone reading any literature on respirometry about how do you prepare the sample and what is happening to what happened in vivo? What am I changing in my assay? And so the bottom line is some of these considerations and others ultimately mean by the time you get your tissue in the respirometer to measure oxygen consumption, there's a very good chance that any post-translational modifications to an enzyme that you're targeting with your substrate, let's say pyruvate for pyruvate dehydrogenase, just a simple example, those post-translational modifications, and what I mean by that is, for example, phosphorylation, could very well have been reversed. Well, why? Why would it be reversed? Because the stimulus of exercise is gone. And because we have now been a couple hours of this sample not frozen, right? And so what can we do as a field to optimize methodologies is another debate, but there's lots of ideas. And quite frankly, it's informed from prior methodologies of things you can add that I don't know if it'll work, but there are people trying it, and we've tried it a little bit, to ultimately allow us to have a higher degree of confidence in the use of respiration to capture post-translational regulation of enzymes resulting from exercise. And that is rarely discussed in an exercise-based study that used respirometry. And again, I don't mean that as a criticism, I mean it as a flag for people to think about. When you see a study saying respiration has changed, I don't know what that means in vivo. What substrate did you use? And how did we truly trap that pathway during the in vitro assay that reflects the degree to which the pathway of interest was regulated in vivo? So there's no definitive answer to give other than I encourage everyone to really learn the regulation of skeletal muscle metabolism, mitochondrial metabolism with classic theories, and look at the method sections of any paper you read to guide your own interpretation independent of what's being said. And I think very quickly, last comment I'll make is you'll see there's doubt. There's doubt in any technique we use, and that doubt is good because it guides our interpretation and makes us realize what we can do more of for your point, which is the measure of mitochondrial function.
And you brought up like substrate utilization and measuring the gas exchange. And do we know anything about like what fuels we're using during exercise and how it impacts these outcomes of respiration? Like, do we see, lower intensity exercise, higher fat oxidation as measured through gas exchange? And then we see these improvements in the respirometry data showing that when we give these fatty acid substrates now, they're using them, or the mitochondria is using them better to produce ATP. Like, is there any relationship between substrate utilization and respiration with these different substrates?
Yeah, specifically for respiration, not the classic methodologies, right? Would be your question. I need to bring myself up to speed on the last couple of years of research to answer that question. I remember several years ago wondering if anyone had done a respiration study to answer your question specifically. Like, has anybody actually just revisited the classic studies of exactly what you said, comparing whole body measures of carbohydrate and fat oxidation to a respiration-based assessment of a carbohydrate substrate-induced respiration or fatty acid-induced respiration? In other words, repeating the classic literature without respiration. Right. These studies were absolutely done with other techniques, radioisotopic fuels, and combined with all the things we already said, not to repeat myself, targeted enzyme assessments and so on. And so we already know the answer is yes, the whole body is related to muscle-specific changes in response to acute or chronic exercise, changes in these enzymes. But did people repeat those questions, now substituting the methodology for tissue respiration? I don't fully know that literature in the last few years, so I can't answer your question. But when I was looking into it a few years ago, there was at least one study, and I think I know the group. I won't name them because I need to look it up, that did that. Actually, that was not too long ago. It is coming back to me. And I honestly can't remember the conclusion. So I guess I'm not prepared to answer your question for the respirometry. But what I will say is when I was looking into several years ago, it was surprisingly few studies had been done to answer your question, substituting classic methodologies with tissue respiration. So I was left very much thinking about, well, if anyone were to do that study, personally, I think, again, I'll use that pause button. We need to hit pause and think very carefully about how to trap the pathway of interest in its activated state that existed in vivo before we ever do that study. Of course, that takes a lot of optimizing, and I suspected that would happen as a field before such studies would be conducted. So that's where my thinking left. And I'm sorry I can't fully answer your question about did they end up seeing respiration led to the same conclusion of a correlation So whole body oxidation is classic methods. What I will say is bigger picture. It goes to show that whatever was or was not shown by whatever study that's not coming to mind, to answer your question, we cannot forget the classic literature. Respiration is not a better method just because it's more popularized. I almost said the word newer. It's not newer. It's been around for almost, we're coming up to a century. But I think one way to use your question as an opportunity is to let students know that even if that study was done, or even if it wasn't done, don't just read respiration literature. Go back to the classic studies, because they're all tools and they all have their limitations. And I think we've addressed the value of respiration, but also the uncertainties of how to align it with what we know with metabolic control theory. So I know that doesn't fully answer your question, but I can say this: other studies outside of the exercise literature do not always find a connection. So other disease states, I shouldn't say other disease states. Exercise is not a disease state. In certain disease states, you don't always see a change in, say, pyruvate-supported respiration, despite not necessarily whole body gas exchange, but despite other measures of carbohydrate oxidation. And I know that's a broad description, and I'm not saying what those measures are, but no, sometimes you don't see the expected change in a respiration based assessment of, say, pyruvate oxidation in certain tissues from certain diseases, when you know other methods are saying it is changing. And that, to me, is exactly the type of flag that we need to carry over to the exercise literature. And
yeah,
again,
sorry, I want to interrupt you quickly. Like, would this have to do, or like, in my mind, I'm thinking disease states, how that changes RER, and then what that would mean from what you're saying, and maybe a disconnect. Like, if someone has metabolic syndrome and they have a really high RER, insulin resistance, type 2 diabetes, but then poor mitochondrial function, if we were trying to say like, oh, substrate utilization is connected to our mitochondrial function, then we would think that these pyruvate substrates would do really well in the mitochondria of their muscles, right? Is that where the disconnect would be?
yes, along those lines. And so while I can't pull up a citation, a reference to address this, because the insulin resistance field I read now and then, and so I'm not fully up to speed on that literature to say if a study has been done to show what you're doing, although I think it has, and I know which groups to go to because they probably did do it. I think what we can use this as an opportunity as is the factors to consider when searching the literature for those studies. Mm-hmm. And so better yet, let's use a purely hypothetical. And what I'm about to say is not me saying this is what's been shown. Let's say as a student or anyone as a reader wants to answer your question, gets in the literature, and they find a study and that did it. Hey, perfect, there it is. Metabolic syndrome, a measure of whole body gas exchange or something else, or even there's other in vivo methodologies, not just gas exchange, that we keep coming back to for carbohydrate oxidation. And oh, look, it was different. Let's say, I think you used the word higher. So let's say carbohydrate oxidation is higher, and then oh, look, in this respiration protocol tissue, it wasn't higher. It was no different. Is that a technical explanation? Because the respirometry protocol, for whatever, didn't truly capture the degree to which substrate catabolism was being trapped and regulated, or rather being activated in that person, that we somehow reset it in our tissue prep protocol? Maybe. Or is there a biological explanation? And this is very, very important. We spent a lot of time now talking about what regulates mitochondrial substrate oxidation. But the muscle can only use what is given to it and what it takes up. a lot of respirometry-based protocols will either assess isolated mitochondria that have been removed from the cells, or in the case of muscle tissue, permeabilized tissues, where, okay, we have some fibers, but we're going to add some chemicals that strip the cell membrane but leave the mitochondria intact. And why would you ever do either approach? Well, ultimately, by removing the cell membrane, it makes it very easy for any substrate you add to the chamber to quickly diffuse to the mitochondria. The idea being you remove the cell membrane as a rate-limiting step to your outcome measure. In other words, if you truly want to know what the capacity is of the mitochondria, then you don't want anything else prior to the mitochondria limiting it to answer that question. However, in vivo, what limits mitochondrial substrate oxidation could very well be something other than the mitochondria, the delivery and uptake of that substrate. So there it is. Is this a problem in this hypothetical, or is this an opportunity? If the hypothetical again is, let's say you find a study where we know whole body there was a change in carbohydrate oxidation, but in vitro in a muscle biopsy there was no change. Does that mean this is a bad study because they used a preparation that didn't capture, or what is it really telling us? When you consider biology and you consider methodology, you actually see opportunity. For example, one of many ideas of where one can go in reading this paper is go, oh, well, perhaps then the mitochondria were not the rate-limiting step in vivo. It would suggest something between the mouth and the mitochondria in the tissue. What can that be? Well, if you work backwards, glucose uptake into the cell, not captured in respirometry-based assessments of isolated mitochondrial permeabilized tissue. We're removing the cell membrane. When in reality, in vivo, it could very well have been a GLUT4 issue, a glucose uptake issue that is ultimately limiting how much glucose the mitochondria is oxidizing. But that's only because the mitochondria may have been provided with less or more, depending on the data we're talking about. So then it becomes an interesting discussion, right? And this is all classic thinking that I think really needs to be front and center even with modern studies. And often it is, but for my point here is to talk to those learning metabolism and learning, as they're learning the literature, they're also learning fundamentals of metabolic control theory. And that doesn't just apply to enzymes in a mitochondria, it applies to the whole body, the whole cascade of eating a food and oxidizing it in the mitochondria. So what else could it be? What's before a cell membrane, right? I'm actually skipping the extracellular matrix here in the capillaries, but the delivery of substrates through the blood flow. And certainly people with metabolic syndrome, depending on the severity of their condition, there's some interesting theories about there. I'll take it to the extreme, type 2 diabetes, where there's poor perfusion, where they might be, I have to be careful with the terms for those in cardiovascular physiology, it could tease this apart, but for the purposes of discussion, if perfusion to all muscle fibers, the delivery of blood, and therefore the delivery of glucose and indeed oxygen and everything else to muscle fibers is reduced because of a disease, does that mean I should dually affect both? I don't want to go that direction. Could that is not captured. The point is that is not captured in a respiration-based protocol. And so if we start to see disconnects between a whole body measure of carbohydrate oxidation and a tissue measure, we have to think about what we didn't measure outside of respiration. Totally. So I know that doesn't answer your specific question, but I would argue whatever you find and others find when trying to answer your question, if you see something that makes sense, you might go, okay, we're done. Look at that. But if it doesn't make sense, if there's a disconnect, does that mean it's a methodological problem where where in vivo, the mitochondria were actually changing their degree of carbohydrate oxidation, but the method didn't capture it? Well, how do you resolve that? Until the field refines itself, what other assays can we do? And that we can learn from classic literature, which we talked about at the beginning. or is it a biological explanation? And so why am I going on and on about it? Because I think, again, I mean this constructively, when I think a lot of publications feel limited in how much they can integrate their findings with the literature in the discussion section, and how much detail they feel comfortable truly unpacking about technical limitations, because we want to write concise discussions. So my point with that is, how is a student ever to think about all these things if the paper itself is not discussing these things? And for that, it really does come down to thinking for yourself, but teaching yourself about whole body to tissue regulation of substrate metabolism.
Okay. So bringing this back down to earth, do you think it's very much so premature to say like one mode of exercise or intensity of exercise promotes greater or better mitochondrial adaptations when it comes to what we are considering function? Like, is high intensity or lower moderate intensity exercise better, or what for improving mitochondrial function? Like, are you of the opinion that it's too premature to say that?
I hate saying yes and no, and it's yes and no. It's yes in a broad sense, or pardon me, it's yes, it's premature in a broad sense, but no if we apply specifics to it. And I don't mean technical specifics, I mean concepts like specificity of training. There's a lot of evidence that, and I know this is starting to get outside the Venn diagram for your stuff, or for your question, but
that's okay. You can take this wherever you want to go.
Yeah, I know what I want to say. I just want to make sure it doesn't go down a too detailed path here. But essentially, I think another way to view this, and I hate reversing questions, but I think one lesson learned is mitochondria are there to adapt to the needs of the cell. They're incredibly plastic, incredibly adaptable. We see that in all sorts of disease states, and allow me to go up and then down. It's amazing when we study, like as a lab, when we study exercise, how exercise affects mitochondria, and then how mitochondria respond to disease. It's funny how I worded that differently. I'm going to say that the same way. When we study how mitochondria respond to exercise and how mitochondria respond to disease, sometimes you see the exact same thing. We have seen in at least 2 muscle weakness disorders, mitochondria get better, where pyruvate respiration goes up in a disease. And yet, if we did that with an exercise study, we would conclude what? Exercise improves mitochondrial function, pre-redox station. So are we prepared to say these muscle weakness disorders make mitochondria better? So I'm going to use that as an example, okay, to answer how difficult it is to answer your question. And your question is critical, and because it's such a popular one. Or is it too soon to say there's an optimal exercise intensity to improve mitochondrial function? Well, I think why would a disease improve mitochondrial function in muscle? Why would pre-redox station go up? Well, 2 ideas, and come back to the exercise to see how it's difficult to answer your question, which is why the answer is yes, it's premature. The substrate we're targeting may have been a conscientious choice, if I can use the word conscientious, a programmed choice by mitochondria to switch to that fuel for some unknown reason. Okay, is that an improved function, or simply a reprogramming to use a fuel for a particular reason? Yes, we get ATP from carbohydrate and fat, but carbohydrates and fats are also used for other purposes beyond ATP. We use fats to make membranes. We use carbohydrate as the backbone for DNA. Like, where does the ribo and ribonucleic acid come from? It comes from glucose. So this becomes really interesting. And I think when we start to look at disease literature on how mitochondria switch the fuel of choice, and we're starting to go down mental pathways of, well, is that a good thing or a bad thing? Or is it just an intentional reprogramming for a grander purpose? I think we can take that thinking for the disease literature and come back to the exercise. Well, what's the best exercise to improve mitochondrial function? Well, the classic dogma is if respiration's up or an enzyme's up, it's a good thing. Well, I don't know if it's good to what end? It's good to run longer. It's good to be stronger. Yes. But I think to say what is optimal in terms of the intensity to initiate or to lead to improved function, guides our thinking in a way that loses opportunity for more insight. And what do I mean by that? What I mean is I think mitochondria, I think it's very clear, studying many different diseases and conditions as a field, not just our lab, that mitochondria will respond to the stress imposed on it by the cell. Mitochondria will adapt to meet the needs. So if you want to run longer, talk to a coach, honestly, about the end output needs to be, the end goal needs to be what is considered, not the mechanism to reach that goal. And what do I mean by that? If you want to run longer, well, what do we know in terms of the optimal exercise training program to improve your time, to improve your strength? If we start going, okay, yeah, I know that, and that's what we've always done, but now I want to be even better at training by thinking about my mitochondria. Okay, well, separate from that question is a question of, well, what training would be best to improve my mitochondrial function? Presumably because that answer will allow me to run longer. I don't think that's the key point. I think the key point is, what do we know makes you run longer? Because, and the mitochondria will have a role to facilitate that. The risk that I'm concerned about is if, if people are saying, and tell me if you think this is happening, I don't know, but my sense is that if people are like, oh, I got to do this training because it's best for my mitochondria, well, that's not the important question at all. The mitochondria will adapt to the mitochondria will facilitate I'm jumping ahead. If you engage in the optimal training to achieve your goal, which is presumably not mitochondria, it's to run longer or to be more fit, it becomes an academically interesting question of how mitochondria are part of that process. But if we make mitochondria the end goal, I'm not so sure that will always facilitate the true end goal.
So I actually just, like, from my perspective, it's no, I want to improve mitochondrial function just based on the literature supporting the association between mitochondrial function and disease. I like that you brought that up, because I'm sure a lot of people have a performance outcome in mind. But from my perspective, it is like, okay, I'm not doing a time trial. Like, I'm not trying to run longer, but I do want to improve my mitochondrial function. Yes, for like health span and disease prevention.
My mind was on the exercise literature. So from a health perspective.
Yes. So how do we use exercise to get that health outcome? And and then what does that tie back to our mitochondria? So is there like an optimal exercise intensity that we can be doing? Because I've heard this, like, this is a very simplified theory that I've heard, and you can give your opinion on this, but like, because of the differentiation, which we haven't actually got into, and I'd like to, between like content and function, like, would it be, again, this is really oversimplified, but would it be like high intensity interval training for increasing mitochondrial content, and then this moderate intensity long duration for like keeping our mitochondria, like oiling the machine, like humming and hawing? Like, is there an argument to be made for that rationale for why we should be doing these polarized training with moderate and then high intensity, but they're targeting different aspects of the mitochondria?
Yeah. Okay. I understand where you're coming from now. Not performance-based, but so
I mean, it's all related, but yeah.
Sorry, it absolutely is. Your second question can be answered without considering health, but do you want me to get into the content function or talk about the first part about the health?
Let's go content function, and then we'll go into maybe what that has to do with health.
I mean, it's an attractive idea that, should we do HIIT training to get more mitochondria, and then lower intensity training to, I'll just say it, improve the mitochondria that we have. it's one of those things where there will be truths to it, and also, as you said, oversimplifications. I think what we know grossly in the literature, from mitochondria to even just health and fitness, and this is also a contentious statement because I can refute it, is that doing a variety of exercise, doing physical activity and or exercise, because I know those can be broken down, of a variety of intensities and durations is probably optimal for health. Okay, so just doing one or the other, I mean, you're still going to be really, really healthy. There's plenty of people, this is anecdotal, I mean, that never do high intensity exercise but they work all day long, right? Does that mean that they have better mitochondria, but not as much as somebody that doesn't work all day long and just does a 30-minute Thursday night HIIT workout? Well, that's a very interesting question, and I don't think the literature is there to answer. However, the part that keeps sticking in my brain is the notion that HIIT exercise may not improve the function of the mitochondria themselves. It will only increase content. Right. And again, that doesn't make any sense, because when you look at
I'm on your page with this, just so you know.
Yeah. I think I know what's driving. I think, and I'm not criticizing you. I think I know what's driving that. And it's probably, I don't remember the studies anymore, but what was the marker of mito content and what was the substrate used to measure oxidation? It all comes down to methodologies, because there's plenty of classic evidence that if you engage in HIIT training, you're going to, I'm going to now transition the word improve mitochondrial function to alter substrate regulation. What that means is shift, reprogram the mitochondria to use a different blend of fuels that are perhaps optimal for endurance. Okay, or optimal for, again, it depends on what's being done here, optimal for the type of activity being done. So let's go from general to specific. There's other evidence beyond respiration that repeated high intensity exercise, okay, so HIIT training, will improve the ability of a muscle to respond to a subsequent challenge, such that they will do a better job at maintaining the energy charge of the cell, the delivery of ATP, if I could put that broadly, the next time there's a bout of exercise. Is that really only because we just have more mitochondria? Very, very, very unlikely when you consider classic literature of the regulation of these enzymes and so on. So why that idea is being put out, I would argue we have to dissect down the methodologies of, again, what substrate was used. Mm-hmm. If a paper proposing that used, I don't know, let's say glutamate is a very common substrate for respiration. I have no idea if that was used. Well, what does glutamate reflect? I mean, ultimately it can come from glutamine. It's an amino acid based substrate. It doesn't just reflect that. But did the study, so to answer your question, again, I'll give some suggestions for a reader. Go to the methods and see, did they compare pyruvate to fatty acid? If a conclusion is that HIIT training does not improve mitochondrial function or even makes it worse, well, what substrate was used? Did they compare the two major fuels, carbohydrate to fat? And if they didn't, then no wonder. No wonder there was no improvement.
And does this also come down to like fiber type? Because if we're changing the mitochondria, like the adaptations are specific to like type 2 fibers. But then if we were doing moderate intensity, would we be targeting adaptations in our type 1 fibers?
Excellent question. Very, very little literature on that. We need to do a much, much more in-depth job on that. I think there's huge potential for what you're saying to be true. There's certainly studies out there that have compared, in rodents at least, muscles that are pretty well devoid of a type 1 fiber, like say a white gastroc, to muscles that are rich in type 1, depending on whether this is mouse or rat, like a soleus. And actually, I'm more familiar with that in the insulin resistance field. And absolutely, there's muscle-specific responses or resistances to disease states like that. Carrying that over to the exercise literature with this question of, would HIIT training change mito content and not function, and would lower intensity exercise change function and not content? Absolutely great question. I don't know if anyone's done that study, and I haven't looked. But it's difficult to do that in a vastus lateralis biopsy from a human. There are ways to go about it, but that's a mixed fiber type, and there's only so many muscles we can biopsy. There are groups motivated enough to try to tease out the type 1 and type 2 fibers and so on from a muscle biopsy and try to do assessments on that. It's difficult to do function on teased out on isolated fibers. It's more markers of protein content and so on. But there's got to be a study that's done something like that that's escaping me. And so I would look for that sort of literature. Certainly in the rodent literature, it's there. So I know I'm still not really fully answering your question about, what's the relationship between mito content and mito function in terms of adaptations to exercise, and should you do HIIT to get more mito was your question, and should you do lower intensity? I think at the end of the day, it's safe to say there's not enough proof to answer your question. There is merit to that thinking, because HIIT training is known to induce very early increases in mitochondrial enzymes. Now, that doesn't prove you have more mito content, but it's a marker of it. And it can be very rapid, like within a few workouts, where you start to see some increases. Does that mean it's faster than moderate intensity training? There's actually not a lot of time course studies comparing HIIT training to endurance training. But if you just look at the endpoint, after several weeks, well, there's several studies that actually suggest the endpoint on mito content markers is the same. And then there's studies that suggest it might not be. And so it becomes impossible to fully answer the question when you have that discrepancy. So how do we go about answering your question if there's discrepancies or different findings? I think it comes down to, again, opening the papers and dissecting the approaches and methods and seeing what more we can do. Very, very likely, HIIT training is going to, if we're going to use that term, improve function. HIIT training is a metabolic shock. Okay, exercise of any intensity is a metabolic shock to muscle, but a high intensity contraction threatens the minimal ATP reserves even more rapidly. And so, yes, the muscle will adapt very likely by getting more mitochondria to make sure that threat is mitigated the next time. But it just seems so unlikely that it's just not going to make its rate-limiting enzymes more responsive at a post-translational level. And I think that's where the question needs to be re-examined with not just respirometry. I think these studies, there's huge opportunity to go back and do more targeted pathway assessments before we say HIIT training doesn't improve mitochondrial function. I just don't think there's enough literature using enough methodologies yet. But it's from a health perspective, can you be healthy if you just do HIIT training? Can you be healthy if you just do endurance training? Of course you can. Is one better than the other? Well, probably, but I think we're starting to split hairs. It's academically interesting,
Right.
But yeah.
And so bringing in the health perspective, what is the relationship between mitochondrial function, mitochondrial content, and then health and disease?
Yeah, very simply. Well, there's a simple, yeah.
It started off simple in your head there.
Well, I'm getting caught up in recent discoveries about the early effects of disease and versus
Right.
long-term.
No, you can explain that too, like maybe these chronic stressors of disease versus.
Well, a very important question is whether mitochondria are becoming dysfunctional in disease. And again, I think the disease that most exercise people think about is diabetes and insulin resistance, but more and more we're thinking about, the field's thinking about other muscle weakness disorders like cancer and so on. And, I'll start with the disease to answer your question. Depending on the time point, depending on the stage of the disease, and depending on the muscle type being examined, time and muscle type absolutely changes the answer to your question. As one example of many, and we're not the only ones to do this stuff, but we just published a study in a mouse model of cancer-induced muscle weakness. And early on, we see in the diaphragm and the quadriceps muscle weakness. There's less force production very early in cancer. And when you look at pyruvate-driven respiration as an index of glucose oxidation using respirometry, with its advantages and limitations, depending on the muscle you look at, you can actually see lower respiration. But what's really interesting is if we then go to a later stage of cancer in this mouse, and then we look at the mitochondria, pyruvate respiration, it's actually up. So hang on here. How would early stage cancer, and by the way, it's a colorectal cancer, nowhere near the muscle, early stage cancer, do what you expect, which is lower respiration, at least off of pyruvate, but then let cancer go long enough and the tumor's getting much bigger, nowhere near the muscle, and suddenly the muscle pyruvate respiration goes up. So what's the answer to the question? Your question, I can pick a time point to answer your question. Classically, everybody wants to say, well, diseases cause mitochondrial dysfunction. Well, actually, mitochondria are highly responsive. This is the key thinking, I think. Mitochondria will help the cell as much as it can in response to a metabolic stress, and not just metabolic stress. The question becomes, what stressors are cancer imposing on the muscle that the mitochondria see, I'm going to put this simply, see an opportunity to help out with? Could it be that early during cancer in this mouse model, there was a metabolic stress? There was certainly we saw lower pyruvate respiration. We didn't measure fat oxidation. I don't know if the mitochondria were simply choosing to use a different substrate. That's not a dysfunction. That's an intentional reprogramming that we did not rule out. But if it was an impairment, and what we mean by impairment was a lower ability of mitochondria to meet the energy demands of the cell, then how do you explain with that thinking that later on in cancer, the mitochondria pyruvate respiration went up? It probably, what we think may have happened was there was a truly an impairment in mitochondria early on, and they sensed this, and the cell sensed this, and quickly reprogrammed mitochondria to compensate. Now, we suspect, we didn't measure it, if you go long enough, that's going to get worse. But had we only picked one time point, we might say cancer is awesome for muscle mitochondria. Cancer improves mitochondrial function. Right. So what's the relationship between disease and mitochondria is your question. We have to be very careful answering that question depending on the time point and muscle type being assessed. In insulin resistance, I'll leave this as a point to consider. If we read the work of Deb Muyo, Daryl Neufer, and others, they've really challenged the classic approach of measuring mitochondrial function in diabetes and insulin-resistant muscle, which generally uses the term capacity. Capacity for fat oxidation is lower in mitochondria from muscle with people with type 2 diabetes. Well, actually, there's a lot of evidence to suggest that we will never even tap into our capacity for fat oxidation. Right. Especially if somebody's sitting quite sedentary, they're not even limited by it. It's like saying, the analogy would be you have a Ferrari and you're sitting at a red light, or you're driving at 20 kilometers an hour, and your ability to drive at 20 kilometers an hour is going to be impaired if you lower the max horsepower of the engine. Well, why would it be? So that's the dogma in the insulin resistance literature has been, well, we have reduced capacity for fat oxidation. Why is the word capacity being used? Because capacity measures were made. Maximal fat oxidation in mitochondria, or the content of an enzyme, and there's other assays as well. So what's the relationship to disease and mitochondrial function is your question. For those interested in that question, especially applied to insulin resistance, take a very careful look at whether or not measures of fat oxidation were made in the context of the daily life of the person. The low metabolic demand of sedentarism, Is the mitochondria truly providing an insufficient amount of ATP to the cell? Well, most people in insulin resistance aren't even arguing that there's a disruption in, if we're impairing fat oxidation, we'll have fat accumulate in the muscle cell. And there's a lot of evidence for that, but there's a lot of countering arguments for that as well. I think in all likelihood, mitochondria absolutely do fail in a lot of diseases, but I think there's an opportunity for more discussion on how much of that change was truly a dysfunction or a reprogramming as a stress response that is part of a larger umbrella. What if there's a reason why mitochondria switched away from fat? What if that's not a dysfunction per se? It leads to a negative effect, if this is happening, of more fat in the muscle. But what if that's a consequence not of an error, but actually maybe we need to revisit lower fat oxidation in mitochondria in these diseases with a question of, well, does that mean ATP levels are dropping in the cell? Does that mean there's not well, no, that's not going to happen. It's still needing energy. So where is it getting its energy from? Or where's the cell getting its energy from? There could be an intentional reason amidst the failure. And I'm worried that we're not fully understanding how mitochondria adapt to the stress of insulin resistance through an intentional reprogramming separate from an actual failure and dysfunction, because that could lead to whole new therapeutic targets.
Right.
So yes.
Because like a theory is that like mitochondrial dysfunction then downregulates fat oxidation, then we get this lipid accumulation and that impairs insulin signaling and now you're insulin resistant. But you're saying that that might be disconnected, like the mitochondrial dysfunction, that might not actually be a leading cause to that cascade.
It could be a leading cause, but I don't know if that should be called a mitochondrial dysfunction until we do more research on whether or not the mitochondria were triggered to move away from fat for some other reason that led to a negative consequence.
Okay.
And we're seeing that kind of thinking in the cancer biology literature, where a mitochondria may turn itself on or off regardless of the fuel or switch from one to the other. Wow, the exercise or insulin resistance literature would immediately call that, and I don't mean to be I don't mean to be rude with it, immediately call it, wow, that's a dysfunction. And it's like, but in the cancer literature, it's, the question is becoming, well, why did it choose? Was it an intentional program away from a substrate because that substrate was needed for some other purpose beyond ATP?
Mm-hmm.
I think there's huge opportunity to take that thinking over to the exercise and insulin resistant field. Just because something's down in mitochondria, that's the thing. Directionality is assumed to be directionality is immediately interpreted as a good or a bad thing. If it's down, it's bad. If it's up, well, no. I think there's opportunity to take what you just said about if fat oxidation is down, it's leading to more lipid accumulation that's driving insulin resistance. There's a lot of evidence that that's happening, but is it arising because of a mitochondrial dysfunction or mitochondrial reprogramming for some other purpose would be how other fields approach the question. And I think there's opportunity when we read different fields to carry some ideas over.
Yeah, I've actually went through some of Brett Goodpaster's research, disconnecting if you lose weight through just caloric restriction versus exercise without weight loss, you can improve insulin sensitivity in both, but you only improve mitochondrial function when exercise is used in the intervention. Yeah. So it does throw a wrench in the theory. And it's all very, very interesting, but
very much so. Very much so. So I know I might not have given the hardcore answers to questions, but I think these are the things to think about. For
sure. No, I really appreciate your perspective. It's given me a ton to think about and how I'm going to approach the literature going forward. But as a last question, because we've been going for a really long time, this has been an awesome conversation.
Oh, sorry.
But no, don't apologize. This is amazing. As a last question, do you think that there are any or non-invasive or more clinical assessments of mitochondrial function? Is there any way that just anyone could kind of determine if they have good, I won't say good or bad, but just mitochondrial function. I'll just use that term for simplicity.
There's definitely clinical measures, and they have they have their advantages, and then what you can and can't measure with them. But then I heard the word anyone. Can anyone measure? That's a challenging one. That's a really important one. No, I often my mind often comes back to blood lactate, and just at the end of the day, it's got to be strongly. It is strongly influenced by the degree to which mitochondria are oxidizing glucose. Just because lactate is up doesn't mean glucose oxidation is down. What it likely means is more glucose, more pyruvate was being provided to the mitochondria than it could use. But even that is nothing's 100%. So, people with blood lactate analyzers, be very careful saying high lactate means I have a mitochondrial dysfunction or so on, but it gives you enormous insight into whether or not mitochondria are using all the glucose being provided. But I don't know how to unpack that into problems with mitochondria or not. In fact, you can't. In terms of clinical research or diagnostics.
Wait, before you go into diagnostics, can we get more into specifics of lactate? Like if you were to measure lactate during, would you recommend like a certain protocol and then measure lactate at certain points to determine like, oh, is this, and maybe not even comparing it to like a range or something, just being able to do like pre-post in yourself. If you were to see like, oh, are my mitochondria improving because my lactate is now lower at a given intensity? Is that what you mean?
I would never feel comfortable saying a change in lactate must mean my mitochondria are different. Rather, a very strong possibility is there was, again, I know this sounds technical, but it's probably one of the most technically accurate answers. Lactate is produced when pyruvate does not enter through pyruvate dehydrogenase. So if lactate's up, does that mean PDH was turned off and the mitochondria just weren't using carbohydrate? No, not necessarily, because if you're filling a carboy of water, you got a big keg with a tap, right? And you're putting water in, which is pyruvate in the mitochondria. Eventually it's going to spill over. If there's no lid, that's lactate.
Yeah.
Now, if you turn the tap on and the water comes out, that's pyruvate going into the mitochondria. Okay, so you should see less spillover, less lactate.
Right.
But if you turn the main tap on and pour lots of water in, even though that tap is open, you could still have a spillover. So you might look at the spillover and be like, oh, the tap's closed. You might look at lactate and be like, oh, the mitochondria is not using pyruvate. No, the rate at which pyruvate was being delivered may have ultimately exceeded the rate at which mitochondria are using. So if mitochondria are dysfunctional, or if you train, right, and your blood lactate response to exercise goes down, which it should, does that mean your mitochondria are better?
Yeah, well, right. Because it just means that you're like now using different fuels. Because you could be using more fatty acids and now you're not even creating as much pyruvate.
Excellent. And vice versa, if a person had elevated lactate relative to, I don't know, another person or, themselves one day or the next, if there was day-to-day variability, like, oh, wow, today I have a higher blood lactate when I exercise. Does that mean my mitochondria are shot today? No, it's relative to what's being provided. Yeah. So no, I would not use lactate as a standalone interpretive tool to conclude what's going on with the mitochondria as a good or bad. It's rather mitochondria are an influence to that measure.
Right. But you did bring it up as an indirect, like non-invasive measure of mitochondrial function. So what were you meaning by that? Maybe I might do wrong.
No. So I don't think there's any standalone clinical measure anyone can do. To answer your question. Okay. So what are markers that can be done that by anyone if they had access to basic equipment that might give that. So let me put this differently. What I'm saying is blood lactate is very, very likely influenced by the degree to which mitochondria are oxidizing glucose. And therefore blood lactate will be heavily influenced by that. However, blood lactate on its own does not allow us to make a good or bad conclusion about mitochondria. And also, there's much more going on with lactate now than just whether the fate of pyruvate, because lactate itself, once in the blood, can be taken up by so many different tissues, cleared by the heart, cleared by the liver, etc. And lots of literature about lactate being a mitochondrial substrate, which still in active area of research. So,
like if you had high fat oxidative capacity and then had higher MCT transporters, then would you expect to see lower blood lactate levels because they're now substrate for the skeletal muscle?
Many theories, there are key parts of theories that would predict the answer is yes, but there's more to it than that to really say definitive yes. Okay. I think there's a lot of classic literature that if you have more MCTs, again, it depends on the tissue you're talking about and the isoform of MCTs, but MCTs can be bidirectional, right? So if you have more MCTs, you might not just take up more lactate out of the blood, depending on the tissue you're talking about, you might actually release more. And so that's why blood lactate on its own it's like saying, looking at your bank account and saying, am I making more money? Well, if your bank account goes up, oh, I guess I'm making more money. Well, what if you spent less? The problem with blood lactate as a standalone measure, I was just saying that it is an easy measure to make that is influenced by mitochondria, but on its own is not a flux measure. A flux measure is how much went into the blood and how much came out. And there's no easy way for anyone to do that.
Right. Sorry, you probably weren't expecting me to hammer in on this question.
No. It's good. You're asking the most important questions. And again, there's no perfect answers, but I think that's where these conversations are good for factors to think about when, well, if you're a scientist wanting to come up with that simple diagnostic marker. Let me tell you, there's massive investments in industry on trying to find an in vivo biomarker of mitochondrial function. If function is defined as the ability to produce ATP, and we're talking neurological disease, industry startups, cardiac is a big one. The cardiac diagnostics field and pharmacology field is really, really trying to invest and discover a non-invasive measure. Or let me say it, you can't biopsy a heart, right? So how do you know if a person's heart mitochondria are better? Well, how do you do that? So they will use blood lactate as one of many measures. They'll use other measures of fuel in the blood. They might even take a peripheral muscle biopsy and cross your fingers that whatever's going on in the heart might be reflected down here. Some studies will go so far as to take a left atrial appendage or a right atrial appendage from somebody getting a bypass surgery. So if you want to know what the invasive studies that will measure mitochondrial function in human heart take a small piece of the heart during heart surgery. So in other words, if somebody's getting a bypass, coronary bypass, there's a little piece of a heart called appendage you can actually cut off and assess its function. And people are doing that. Wow. But you're not going to do that diagnostically.
Yeah. So now to the diagnostic portion of your answer.
Yeah, and very, very briefly, MRS, magnetic resonance spectroscopy, sometimes called NMR, nuclear magnetic resonance. Essentially using this is not the right term, but magnetic resonance of phosphate, of protons. If you apply a magnetic field to tissue, certain ions will actually resonate, and that resonance can be picked up. And I'm not an expert on this, so there's much more to it than that. But what that means is you can detect one of the phosphates on ATP with NMR, and you can detect phosphate on phosphocreatine, and you can detect other metabolites by looking at essentially the resonance of a key atom on the structure of that substrate. This is really useful because now you can look at the rate at which ATP or phosphocreatine is cycling. And it's a powerful technique. And so that's been used a lot in different disease states and before and after contraction. People have done that with an NMR magnet around the forearm, around the calf, and so on. That's been around for a while now, and it's really, really insightful because you get in vivo data without a biopsy. When you see a change in the turnover of ATP or phosphocreatine, what caused that change? Was it change in carbohydrate oxidation, change in fat oxidation, change in a certain enzyme? It doesn't tell you that. So every methodology, what does it tell you and what does it not tell you? So it'll tell you grossly what's going on with energy provision connected to mitochondria, to put it loosely. But if you want to now know what caused that change, there's no good non-invasive measure.
Mm-hmm. And I sent you some of the papers on the fat max and how it might be a functional assessment of mitochondrial function, or of our mitochondrial capacity, whatever word we want to use. What do you think of doing like some sort of fat max test like that and using gas exchange?
For exercise studies, or are we still talking clinical diagnostics, or?
Yeah, like could you measure your mitochondrial function indirectly by doing something like a fat max test?
No. Mitochondria will absolutely contribute to the maximal rate of fat oxidation if mitochondria themselves are rate limiting. And I know it's never simple, right? But it becomes a question of what are all the factors that regulate maximal fat oxidation or fat max.
Right. Okay.
Like lipolysis of adipose tissue, delivery of fat in the blood, and then uptake at the cell membrane. And remember, a whole body measure isn't necessarily just driven by the muscle oxidation. Will be obviously the predominant one, but other tissues that could be turning over the fat as well, less so on that. And then eventually the mitochondria. So, no, you can't just use gas exchange to measure mitochondrial function, but if someone's mitochondrial function or regulation changes, it can be impacting fat max. But at the same time, if altered regulation of lipolysis, like catecholamines or other regulatory factors have changed, you could see a change in fat max that had nothing to do with the mitochondria.
Right. No, I appreciate you bringing those up. Yeah.
Yeah.
Okay.
Yeah. Okay.
This has been amazing. I appreciate your time so much. I had so much fun during this conversation. Would you like to leave off with saying where people can find you? You're pretty active on Twitter. Maybe you want to share your Twitter handle. Anything else you want to share before we sign off?
Oh, yeah. I mean, if anyone, I'm always open to emails from anyone with questions. And honestly, I always learn from them as well. I get random emails from people after conferences, symposia, training sessions I offer on mitochondria. And I might be a little delayed sometimes replying, but just email me, cperry@yorku.ca. I don't even know my Twitter handle. Chris
Okay,
Perry.
I'll link your
Chris
Twitter.
Chris Perry Mito. Yeah, absolutely. And we don't have to talk as long as we did today.
Well, okay. Well, have a great rest of your day. And again, just so thankful for this
Thank you, Christy. Thank you for inviting me. And it was a great discussion. You're asking great questions. Thank you.
Appreciate it.